Hi students, the last class we talked about the cloning mechanism, how the cloning animals produced. That's the mechanism of cloning. Now we have to study something about the advantages and disadvantages of cloning today. Now let's take first one, advantages. Number one, so the cloning animals normally offers benefits for clinical trials and medical research. In clinical trials means we can inject the medicine into the cloning animals and testing whether it is to be applicable for human just actually injection or human administration. So in medical research also we are doing so many research works to find out for example the suppression of particular gene function or just elevating the function of particular gene function. Like that, in all medical research we are using what is called the cloning animals. How in medical field they are being used? Now the cloned animals are used for the production of proteins and drugs. They help in the production of proteins and drugs. That is in the field of medicine. In that aspect they act as bioreactors. Bioreactors where the medicine or a drug or a protein, therapeutic protein just nowadays has been produced. So that is another function, second one. Now it helps in stem cell research. So what is the role of stem cells? How far the embryonic stem cells, the adult stem cells useful? And for that research, for that work, we need what is called the cloning animals. So the cloning animals help in stem cell research. Then what is their role in biodiversity? Now you know, some of the animals are facing the extinction process. They are at the verge of extinction. Suppose you have only a few animals. With these few animals, we can increase the number of that particular individual animal species. So, animal cloning would help because we can produce copies of animals. But ethical issues are there. We cannot just clone animals or clone just actually human beings, but we can clone animals. So, animal cloning would help to save endangered, that is what we have the species. Those animals which are at the verge of extinction, we have only a few individuals for that species. We can increase the number of that individual species by a process of cloning. So these are some of the advantages of cloning animals. Now we have to go for the disadvantages of animals, the cloning animals. Okay, we'll see that. So far we have studied the advantages of cloning animals. Now let's see what are the disadvantages of cloning animals. Number one. Now animal and human activists, that is actually looking into just the various activities proposed by different people. They are looking into find out any just doubt in that one. They are actually acting as critics. So animal and human activists see it, that one, the cloning, a threat to what is called biodiversity. Though its role in biodiversity to develop what is called animals, the endangered animals, which are at the what is called the phase of extinction. That is one aspect advantage. But the same one just actually is a threat to biodiversity. They are saying that what? They alter what we call the process of evolution. When the evolution process has been changed, it will have an impact on the population as well as the ecosystem, the biological unit, where you have the living and non-living factors are formed. So, it also has a disadvantage of just actually a threat to biodiversity, just actually alters the evolution that results in what is called an impact on the population and the ecosystem. The next disadvantage, the process is very tedious. For example, both Jan Wilmot and Campbell, they took nearly 277 eggs and 277 adult cells. Then after fusion, they incubated the cells for about 6 days. Okay, and after incubation they are getting they receive only 29 cells, the fertilized cells, or we can say the cells with the diploid condition. So 277 were taken So we have received only 29 cells with the diploid nucleus. When these 29, that is cells with the diploid nucleus in the excitoplasm, when implanted in different surrogate mothers, the result is only one donor. So it's a wastage method. See, we have taken only 277 egg cells and 277 somatic cells, the adult cells. Then after fission, they were incubated for 6 days and then they were implanted in different surrogate mothers, 29. Out of these 29, only one dolly survived to birth. That's the ratio. So it is a tedious process, not just like it is simple one. And also it's very expensive. It is very expensive also. It's very expensive. Cost is more. Now it causes the animals to suffer. Those cloned animals have some sort of what's called sufferings. Then cloning can lead to the occurrence of genetic disorders. Most of the cloned animals have developed the genetic disorders. These are all the disadvantages. We have to go for some other disadvantages. Now the next advantage, based on the reports, the animal surrogates. So normally animal bearing the embryo of another individual on behalf of another individual. So reports show that the animal surrogates normally manifesting adverse outcomes. Some abnormal outcomes resulted after what we have developed the individuals by cloning method using the surrogate mothers. Now the cloned animals normally have developed diseases and have high mortality rate. The death rate becomes higher. So they have developed disease that results in the death of the cloned animals, which results in increased death rate, that is called high mortality rate. Now if you are comparing the cloned animals and the normal animals, the cloned animals age faster, growing faster, attaining old age quickly. And also they become less healthy than what we have the parallel organisms. So it was reported, that is discovered during the development of dolly by cloning method. So they attain age faster when compared to the normal organisms, and also they become less healthy when compared to the parallel organisms. That was one of the problems noticed during the cloning of sheep dolly. The next one, when the experiments were conducted, more than 90% cloned animals could not produce viable offspring. In one way or other, the produced offspring have some sort of what is called disorders, some sort of what we call diseases. So they are not viable. They soon die. The death rate becomes higher. So more than 90% cloned animals could not produce viable offspring. Though the method is adapted, but only 10% success is there. The remaining 90%, just normally what will happen, the cloned animals will die. Or they are not being produced effectively, viably by the surrogate mother. Now we have one more disadvantage. Now normally cloning, just normally, it might just about the human health, it might compromise the human health through the consumption of what we have the cloned meat. The meaning for that one, the cloned meat when consumed by human beings, they cause some sort of problems. So it compromises, the cloning animals compromise to the human health through the consumption of what we have the cloned meat. They cause some problems in the human body when we consume the cloned meat. This is the last disadvantage. So that's about what we have the disadvantages of animal cloning. We have to go for a paragraph about ethical issues. Okay, now let's see the 8th disadvantage. I'll write down on the board, and then I have to mention about one gene. What is a gene? Knockout gene. Then we'll pass on to, that is, we have the ethical issues. The last disadvantage of cloning animals is this. The cloning might compromise human health through consumption of what we have the cloned animal meat. That means when we are consuming the cloned animal meat, it may cause some problems to the health. That is the meaning for that statement. Okay. So the consumption of what is called cloned animal may cause health problems in human beings. These are all something about the disadvantages. Now a few words about knockout gene. You hear the word knockout in the case of games. The same one here. Now it is a knockout gene is nothing but a genetically engineered organism that carries one or two genes in its chromosome. That genes or gene have been made inoperative. The meaning for that one I here given, it is a process of suppression of gene function. A particular gene function is being suppressed by what is called gene manipulation. That is called knockout, not allowing the gene to express. For example, in the case of mice, there is a gene, what is called MCH. MC for mice, H for hair. This is a gene responsible for the normal growth of what is called actually normal growth of hair in the case of mice. Now what for you are doing that process, suppressing the gene? In order to know the function of that particular gene. So to know the function of a particular gene, we are doing that process what's called knockout. Now in this one, when the gene has been suppressed by incorporating another gene, what will happen? The growth of the hair is affected. So this is the gene MCH for the normal growth of the hair. When this gene is suppressed in the mice, it is not being expressed. There is no normal growth of hair. That is happening. That's why it's called as knockout gene. A particular gene function is suppressed. So this process is normally used to find out the function of a particular gene. By suppressing a particular gene, one can able to know what type of function that gene carries out. That's about the knockout gene. Now the last part, we have to go for the ethical issues, a few sentences. Many people have wrong opinion about that is applications of biotechnology or the products of biotechnology. Now the biotechnology, you know that one has given to the society cheap drugs, better fruits and vegetables, then resistant crops, pest resistant crops, and indigenous cure to medicines. These are the benefits. Along with the benefits, biotechnology has also given a lot of controversy. What's the reason for the development of controversy? The reason is, this is mainly because the major part of modern biotechnology deals with the genetic manipulation, addition, repair, removal of a particular DNA segment. That's the reason why people normally fear about it. So, this is mainly because the major part of modern biotechnology deals with the genetic manipulation. Insertion of gene, addition of gene, just repair of gene, etc. Now, people fear that as we are using genetic manipulation, changing the entire organism, they just normally fear that the genetic manipulation may lead to unknown consequences. Without just appearance, they have led to the development of unknown consequences. Maybe they are thinking that the harmful effects. For example, the major apprehension of recombinant DNA. Apprehension means the impact of recombinant DNA technology is that normally. So, through this one unique microorganisms, either what we have inadvertently, there is nothing but unintentionally, inadvertently or unintentionally. Or deliberately for the purpose of war may be developed. So they develop these microorganisms, biological warfare. So may be developed by people, either just actually unintentionally or deliberately. What is the impact of that one? That result in what we have that could cause epidemics, spreading of disease all over the country, where these organisms have been introduced, the microorganisms with a harmful effect, or we have some environmental catastrophes. Catastrophism occurs because of what we call the introduction of the microorganism that has been made through biotechnology for biological warfare against one country. That results epidemics in that particular country and also causes some environmental, just what we call catastrophes. Either we have the flood or we can change the environment, the climate, etc. That's about environmental catastrophes. Okay, now we have just actually thinking about the risk of genetic engineering. But what is the real one? Okay, let's talk about. People fear about the products of biotechnology through genetic manipulation because they lead to some consequences. So although many consider about the risk of genetic engineering, that's a biotechnology, the risks are in fact very slight, not causing much damage. And the potential benefits, we are getting benefits are substantial. That is enough and more than enough, not causing damage. The benefits are more and what we have, the risks are less. That's about what we have, the importance of ethical issues. So with that I concluded what we have the lessons, applications of biotechnology for biozoology students. We have 2 or 3 pages left for zoology students. So I am taking the next class only for the pure zoology students, the remaining part of applications of biotechnology. Okay, thank you.